Okay, so welcome back to this next video in which we are discussing angiogenesis and angiotherapy. Okay, right, so we're in the process of discussing hypoxia-induced angiogenesis. Okay, so we're assuming we've got some area of hypoxic cells, okay, and under hypoxic conditions, these cells are going to start releasing pro-angiogenic molecules, principally vascular endothelial growth factor A, but also another major one is fibroblast growth factor 2. Okay, and we're going to concentrate on the effects of vascular endothelial growth factor A from now on. Okay, so we've discussed that the endothelial cells have on their surface receptors for vascular endothelial growth factor A, which are vascular endothelial growth factor receptor 2s, which are receptor tyrosine kinases. And we've discussed that when the vascular endothelial growth factor A activates these vascular endothelial growth factor receptor 2s, it leads to major epigenetic shifts in the endothelial cells, okay, which changes the phenotype of the endothelial cells. Okay, so we've discussed that some of the first things that happen are that the endothelial cells increase their expression of endothelial nitric oxide synthase and therefore start secreting more nitric oxide. Nitric oxide diffuses out to the pericytes and causes them to relax, which is what drives the vasodilatation of the capillaries in this local area. In addition, the endothelial cells also start to contract, which leads to the production of gaps in between neighbouring endothelial cells, which then allows fluid from the bloodstream to leave the bloodstream and go into the interstitial fluid, and that creates you an exudate. Okay, right, and those things are going to be important when we come on to discuss diabetic retinopathy. Okay, right, then to actually go to the beginning of the angiogenesis process, what's going to happen at this portion of the capillary that is closest to the hypoxic centre, you're going to start getting the basement membrane being broken down, okay, and that's by matrix metalloproteinases. And one thing that I forgot to say last time when we were discussing matrix metalloproteinases is the reason they're called metalloproteinases is that they are dependent on metal ions, generally zinc ions. Okay, so that's just a little bit of background information there. Okay, so matrix metalloproteinases will start breaking down at the basement membrane closest to the site where the hypoxia is occurring. Okay, so that will produce a hole basically in the basement membrane. In addition, we've discussed that the pericytes will get away from this area basically, so they'll flee this area, they'll pull away from it basically. Okay, and we're just now in the process of discussing tip cells. So the endothelial cell that is closest within the capillary to the hypoxic centre is going to convert into a tip, or also called a leading cell. It's going to undergo this major phenotypic change, okay, where it goes into this shape where it has these filopodia at the front, okay, and these filopodia are extremely important for this cell being able to migrate, basically, and we'll come back to its migration in a moment. What I want to discuss now is why do you only get one endothelial cell becoming a tip cell? Why do you not get more? Why are these endothelial cells not being induced to have the same being induced to undergo the same phenotypic change that this endothelial cell has undergone? Okay, and it's basically because once this one has become a tip cell, it inhibits the others from becoming tip cells using notch signaling. So I want to tell you a little bit about notch signaling. So notch signaling is a signaling pathway that's extremely important in developmental biology, but it's also extremely important in this angiogenesis process here. Okay, so basically in notch signaling you have two major components. You have notch ligands and then notch receptors. So let me tell you about the different notch ligands, then I'll tell you about the different notch receptors, and then I'll tell you which are going to actually be important in this signaling between the tip cells and the other endothelial cells. Okay, so notch ligands then firstly. Notch ligands, there are 5 different notch ligands, and these are called jagged 1 and jagged 2. Okay, and then the other 3 are named completely different things. Okay, so the other 3 are called DLLs. Okay, and there is DLL1, DLL3, and DLL4. Okay, and DLL stands for delta-like ligand. Okay, so the D is for delta, the first L is for like, and then the second L is for ligand, so delta-like ligand. Okay, right. So those are the 5 different notch ligands: jagged 1, jagged 2, delta-like ligand 1, delta-like ligand 3, and delta-like ligand 4. Okay. Now, the other component of notch signaling then is the notch receptors, which bind to the notch ligands. Okay, and there are 4 different notch receptors, and they are just called notch 1 through to notch 4. So there's notch 1, notch 2, notch 3, notch 4. Okay, so those are the 4 different notch receptors there. Okay, right. And basically, cells can put notch ligands on their surface. Notch ligands are not a free molecule. They're not a secreted molecule in the classical sort of way that you would think of a ligand. Okay? Instead, they are something that you can put on your cell surface, and they can bind to another protein on another cell's surface, which is the notch receptor, and then they can have signaling roles in that other cell which has the notch receptor on its surface. So basically, endothelial cells normally express notch receptors. Okay, and the 2 notch receptors that are mainly found on endothelial cells are notch 1 and notch 4. Okay, so let me now show you the notch receptor on the surface of an endothelial cell. So now I'm drawing the membrane of an endothelial cell. Okay, and I think I'll colour this in. So let's colour in the membrane of an endothelial cell here in orange. One of these endothelial cells that is near this tip cell here. Okay, like so. So here is its membrane. And basically now, the notch receptor, whether it's notch 1 or notch 4, is going to be in this membrane. Okay, so this is the outer leaflet, this is the inner leaflet, so this is the extracellular fluid. Okay, and the amino terminus is extracellularly. Okay, then you have a large extracellular domain, which is going to be important in binding to the notch ligand. Then you have a membrane spanning alpha helix, which again I haven't bothered to draw as an alpha helical structure. Okay, and then you have the notch receptor intracellular domain here. Okay, so 2 important domains here. The intracellular domain here, the ICD. So whenever you see ICD in biology, it's always standing generally for intracellular domain. Okay, and then we've got the extracellular domain over here, which will be binding to our notch ligand. So ECD for extracellular domain. Okay, so let's colour these in different colours. So we'll have the intracellular domain in turquoise here. Okay, and we'll have the extracellular domain here in blue. Right, so endothelial cells usually have these receptors, notch 1 and notch 4, which both look like this on their surface. Okay, now, the ligand for these receptors is not usually expressed on the endothelial cells. However, these tip cells, once they have been acted upon by the vascular endothelial growth factor A, and they've been turned into this tip cell, they are now going to start expressing a ligand for the notch receptor. They're going to start expressing one of these notch ligands. Okay, and the specific notch ligand that they start expressing is delta-like ligand 4. Okay, so the tip cells are going to start expressing delta-like ligand 4 on their surfaces. So if I now put the tip cell membrane, which will be close to this endothelial cell membrane here. Okay, so I'll colour in the tip cell here in blue, and because it has now become a tip cell, because it's been acted upon by the vascular endothelial growth factor, it got that biggest dose of vascular endothelial growth factor. And one of the proteins that it has now been forced to express is the delta-like ligand 4. So let me put this on here. Okay, so again, delta-like ligand 4 is a single-pass membrane protein with its C-terminus in the cytoplasm. So this time, this is the outer leaflet of the cell membrane, this is the inner leaflet, so it's the opposite way around to this side. This is the extracellular fluid, and this is the cytoplasm. Okay, and our delta-like ligand 4 just has the important domain here through which it binds to notch here. Okay, so this is the domain which it uses to bind to our notch receptor, which is either notch 1 or notch 4. Okay, right. And then here is the N-terminus of our delta-like ligand 4. So this is delta-like ligand 4. This is either notch 1 or notch 4. Okay, right. Now, so the tip cell has put the delta-like ligand 4 on its surface. It is now binding to notch 1 and 4 receptors on the surface of these endothelial cells here. Okay, the surrounding endothelial cells of the tip cell, basically. And now what's going to happen is when the delta-like ligand 4 binds to the notch receptor, the notch receptor can now be processed, basically, by 2 enzymes sequentially. So what's first going to happen is you're basically going to cut up this notch 1 or 4 receptor, okay? Firstly, you're going to cut it extracellularly here, okay? And that first cut, so this is cut number 1 that's now going to occur after you form this complex between delta-like ligand 4 and notch 1 or 4. This first cut is going to be made by an enzyme known as an ADAM enzyme. Now, there is not just one ADAM enzyme, it's a massive great family of enzymes, but it's going to be an ADAM enzyme which makes this first cut. So what does ADAM stand for? Okay, well, it stands for a disintegrin and metalloproteinase enzyme. So a disintegrin, the A is for A, D is for disintegrin, the second A is for and then the M is for metalloproteinase. Okay, so that's what ADAM stands for. And as I say, it's a massive, great family of enzymes. So it's an ADAM enzyme which cuts there. Okay, so that's the first cut. It's made by a ADAM enzyme, a disintegrin and metalloproteinase enzyme. Okay, right. After the ADAM enzyme has cut the notch 1 of 4 and it's cut the extracellular domain off, now you've got this portion left here with the transmembrane portion and the intracellular domain. Now what's going to occur is an enzyme called gamma secretase, which is actually a multi-protein complex made of 4 separate proteins all bound together, is then going to cut between the transmembrane domain and the intracellular domain. So this is cut number 2. And this is the same gamma secretase that is so important in Alzheimer's disease in processing amyloid precursor protein. Okay, so gamma secretase is going to cut there. And that will produce you now the intracellular domain of your notch receptor, okay, which we'll call the notch intracellular domain. Okay, and this will go into the nucleus of the cell, and basically it blocks the vascular endothelial growth factor from causing many of its effects. Okay, so it blocks vascular endothelial growth factor signaling, basically, and stops the vascular endothelial growth factor A being able to cause the same enormous phenotypic change that you saw in this tip cell here. So basically, the reason you only get a single tip cell is that this one gets the biggest dose, it turns into this tip cell first, okay, and then it starts inhibiting its neighbours and stopping them from responding to the vascular endothelial growth factor A in the same way, so that only one cell is going to actually become this tip cell, and it's through this notch signalling pathway here that the tip cell manages to actually inhibit its neighbours and stop them from also becoming tip cells. Okay, so let's now talk about what the tip cell is going to do. So the 2 key things that it does is it migrates and it proliferates. Those are the 2 immense properties that it's given. Okay, so it's changed in shape this way so that it can migrate, basically. The filopodia here are so that it can migrate. So migrate and proliferate. Okay, so what's going to happen here? Let me get another piece of paper and we'll draw the next picture. Okay, right. So, actually I'll get my picture back again and then I'll... Right. So, basically what's now going to happen is this tip cell is going to move out. It's going to move in this direction. Okay, the filopodia are going to squeeze in over here. So the filopodia are going to start extending out like that, so. And then what's going to happen is they're going to engorge, basically. They're going to get bigger and bigger and bigger and become one great sort of confluent mass here, whilst back here at the behind of the tip cell, you will just gradually sort of get rid of that, okay, so that will shrink. So the way cells migrate is they move a tiny little filopodia into the space that they want to move into, and then that engorges, basically, and the entire cell ends up being over there, whilst they're behind over here just sort of shrinks down, and that's how they move, okay? So the tip cell overall is going to move out this way, and as it does, obviously it's going to leave gaps basically, so it's also then going to have to proliferate and create new cells to fill in the gaps that's left behind, and these cells are then going to become normal endothelial cells, because the instant they're created they're going to be being inhibited by the tip cell in the same way that these cells were inhibited through this notch signaling pathway, and they're going to remain as normal endothelial cells. Okay, so let me draw the picture for this on this new piece of paper. Okay, right, so let's start by putting the basement membrane on. So here's the basement membrane that's still intact, and here's the one where we've got this hole now. Okay, then we'll mark on our tip cell. So our tip cell kind of moved forward now to this point here. Okay, here are a few of its filopodia still. Okay, and now you see it's left this gap here. So what's going to have happened is it's also going to be dividing and producing new cells which will become normal endothelial cells, and they will fill in the gaps here, like so, to create a little sort of stalk basically. You're creating a vascular stalk that's going to become this new capillary basically. Okay, then we've got our endothelial cells back here now. Okay, like so. And at the moment, these new endothelial cells that you've put here, they don't have a basement membrane underneath them. They don't have pericytes surrounding them. They're just the endothelial cells at the present. Okay, but you're starting to produce this stalk. And in fact, these cells, these endothelial cells here that are making this stalk, these are actually known as the stalk cells. Okay, this is still known as the tip cell, but these new endothelial cells that are making the capillary bud or the capillary stalk are known as the stalk cells. Okay, so here in blue is the tip cell. Okay, and then over here we'll still have our endothelial cells intact, and then I'll put a few pericytes on just to complete the picture up. So here is one pericyte, here's another pericyte, and we'll have one down here as well. Okay, so what's going to now happen is this process is just going to continue. Okay, this is step number 1, okay, but it's just going to continue on. The tip cell is going to continue migrating towards the hypoxic center, which we'll have over here, and as it does, obviously it will leave gaps behind it, and it will just make new endothelial cells by proliferating, which will become the stalk cells in its trail basically. So it's the cell that single-handedly creates this entire stalk. It moves towards the hypoxic center, and in its wake it leaves endothelial cells behind that can fill in the gap basically. So it is the essential cell here. It's the one doing the migrating, it's the one doing the proliferating, so it's the incredible cell. It creates the stalk basically. Okay, right. Now, also what you'll be getting is, as it moves forward and the stalk cells are left behind whilst the tip cell continues on, you're going to be getting this, what's known as the stabilisation of the stalk here. Okay, and the stabilisation of the stalk involves producing a basement membrane for these stalk cells to sit on, and also putting pericytes around that basement membrane. Okay, so as the tip cell continues to move forward and the stalk gets elongated, so you can imagine the next step where you'd have now additional endothelial cells here. So I'll draw this stalk longer now. So there's one endothelial cell, here's another endothelial cell. Okay, so if you just imagine this being translated up here. Okay, so here are more endothelial cells as the stalk continues to elongate, whilst as the tip cell continues to move forward. So here is our tip cell here. Okay, right. So basically, what now has to follow is we need to stabilize this stalk, okay? So a process called stabilisation is going to occur, and the two key parts of stabilisation are you're going to get a basement membrane, so you're going to put a basement membrane underneath these endothelial cells, give them some support to sit on basically, and then around the basement membrane you're then going to put pericytes basically. So you're going to turn it into a normal capillary. At the moment all it is endothelial cells in a stalk that has a lumen down the middle. Okay, you want to now turn it into a capillary, so you need to put the basement membrane back and you need to put pericytes around it. Okay, now there are some key molecules that are known to be really important in the stabilisation process. Okay, so firstly let me tell you about how you get the pericytes surrounding you, or at least what is known about how you get pericytes surrounding you. Okay, so it's still a little bit fragmented, our picture of this at the moment, but what we do know is that a molecule called angiopoietin-1 seems to be extremely important. Okay, so let me tell you about angiopoietin-1. So angiopoietin-1 is usually just abbreviated down to Ang1 for short. Okay, and it's still not entirely clear where this molecule gets secreted from, but it is a secreted molecule. Okay, we think it probably is secreted by the endothelial cells themselves. Okay, and then what it acts on is the endothelial cells. So it acts on receptors on the surface of the endothelial cells, and the receptors for angiopoietin-1 are a receptor tyrosine kinase known as Tie2. Okay, so it's just another classical receptor tyrosine kinase like we saw for the vascular endothelial growth factor receptor 2, like we saw for fibroblast growth factor receptor 1. Okay, again, when the angiopoietin-1 binds to the Tie2 receptor, it will dimerize. You'll then get the activation by phosphorylating each other's very special tyrosines on the kinase domain, and then they'll phosphorylate all the other tyrosines in the intracellular domain. So you've got loads of phosphotyrosine residues, and again, that will cause all sorts of epigenetic shifts within these endothelial cells that are in the stalk, basically. Okay, and somehow this seems to lead to the endothelial cells recruiting pericytes to them. Okay, so activating this Tie2 receptor tyrosine kinase seems to bring pericytes around these stalk cells. Okay, like so, which is one of these components of stabilisation. And if I show it on this picture, here's a pericyte coming around these stalk cells here and making it more like a normal capillary. Okay, so I'll colour in these pericytes in red. Right, okay, so angiopoietin-1, or Ang1, is really important in the recruitment of pericytes to around the stalk cells. Okay, what signalling molecule is known to be extremely important in the production of the basement membrane? Okay, well, again, the basement membrane seems to be being produced by the endothelial cells themselves. Okay, so again, it needs to be acting on the endothelial cells, whatever this ligand is. And the ligand that's thought to be extremely important in activating endothelial cells to produce the basement membrane is transforming growth factor beta, or TGF-beta for short. Okay, so this stands for transforming, that's the T, and then growth factor, that's the GF, and it's transforming growth factor beta. Now, there isn't just one transforming growth factor beta, there are multiple of them. However, for our purposes, we'll just group them all under the title of transforming growth factor beta. Okay, right. Now, the receptor for transforming growth factor beta, the transforming growth factor beta receptor, is really quite interesting. It is not a receptor tyrosine kinase. Instead, it is something a little bit more rare, something called a receptor serine threonine kinase. Okay, so it's a receptor with an inbuilt serine threonine kinase domain, okay, rather than a tyrosine kinase domain, a domain that phosphorylates serine and threonine residues rather than tyrosine residues. So let me just tell you a little bit about receptor serine threonine kinases. And once again, we won't be going through the entire signaling pathway. And we'll just be talking about the activation of the receptor, and then we'll say it leads to signaling pathways which lead to epigenetic changes in the cell. Okay, right. So there are two receptor subunits, again, for the transforming growth factor beta receptor. So remember, in receptor tyrosine kinases, you need them to dimerize. Again, in transforming growth factor beta, you need them to dimerize. However, the two components of the dimer are not going to be the same this time. In the case of receptor tyrosine kinases, they formed a homodimer. Okay? In the case of the receptor for transforming growth factor beta, they're going to form a heterodimer. Okay, so let me show this. So, if this is the cell membrane of our endothelial cell, let's say this is the outer leaflet, this is the inner leaflet. So this is the cytoplasm, this is the extracellular fluid. Okay, so there are two different types of transforming growth factor beta receptor, okay, and they're the two molecules that need to dimerize together to make the full transforming growth factor beta receptor. So the one that actually comes first in the story is the type 2 transforming growth factor beta receptor. Okay, so I'll just draw this here. So again, it's a single pass membrane protein. It's got a single membrane spanning alpha helix down here, and here is the inbuilt serine threonine kinase here. Okay, so this is a type 2 transforming, transforming growth factor beta receptor. Okay, right. Now, the type 2 transforming growth factor beta receptor is the one that the ligand binds to. Okay, so here is our transforming growth factor beta binding to our type 2 transforming growth factor beta receptor. Okay, so I'll colour in the transforming growth factor beta in red here. Okay, and it binds to the receptor, causes conformational changes in this type 2 transforming growth factor beta receptor, and this allows the type 2 transforming growth factor beta receptor to dimerize with the type 1 transforming growth factor beta receptor. So only when this ligand has bound can you now dimerize with the other one, which is the type 1 transforming growth factor beta receptor, which also has an inbuilt serine threonine kinase down here. Okay, so this is now the type 1 transforming growth factor beta receptor. Now the type 1 transforming growth factor beta receptor does not actually need to bind the transforming growth factor beta itself. Okay, all that needs to happen is one transforming growth factor beta binds to the type 2 transforming growth factor beta receptor, causes a conformational change which allows it to dimerize with the type 1 transforming growth factor beta receptor. Okay, and then what's going to happen is the serine threonine kinase domain of the type 2 transforming growth factor beta receptor is going to phosphorylate the serine threonine kinase domain of the type 1 transforming growth factor beta receptor. Okay, like so. And that activates the type 1 transforming growth factor beta receptor's serine threonine kinase domain. Okay, so this has now been activated hugely, and it can now phosphorylate serine and threonines on other downstream proteins. Okay, and that's what leads to the downstream cascade. So basically, the type 2 transforming growth factor beta receptor only binds this ligand. Its serine threonine kinase domain is already active. All it needs to bind the ligand for is so that it can dimerize with the type 1 transforming growth factor beta receptor. Okay, so that the kinase domain of the type 2 transforming growth factor beta receptor can phosphorylate a special site on the type 1 transforming growth factor beta receptor, activating that kinase domain of the type 1 transforming growth factor beta receptor, and now it will phosphorylate downstream pathways, proteins of downstream pathways, and cause epigenetic changes, cause changes in which genes are actually going to be expressed in this cell. Okay, so that's how transforming growth factor beta acts on endothelial cells, and again, it amounts to the same thing. Epigenetic changes are what the receptor activation produces. Okay, right. And this transforming growth factor beta, again, it's not totally understood where it comes from, like the angiopoietin 1. It's there, present, ready to act on the endothelial cells when they need to start synthesizing basement membrane, but we don't know exactly how this all fits together yet into an orchestrated process. So we don't know where this transforming growth factor beta fundamentally comes from to signal to the endothelial cells, but it is there, and it is now going to signal to the endothelial cells to tell them that it's time to make some basement membrane, basically. So they're now going to start making basement membrane here and putting it underneath themselves. Okay, so those are key molecules basically that are known to be involved in this stabilisation process, even though the full details of how it all sews together aren't yet established. So we don't quite know what's orchestrating the sudden arrival of the angiopoietin 1 and the sudden arrival of the transforming growth factor beta. Okay, no doubt in the future we will understand that. Okay, right. So that's the stabilisation of the stalk that's being left behind by the tip cell. So now all that remains to say is that what's going to happen is we can go back to our big picture now. We've got this stalk coming off our capillary here towards this hypoxic centre here. And of course other capillaries are also going to be doing the same thing. They're going to be generating stalks in exactly the same way, okay? And these stalks are going to be converging on this same hypoxic centre here, which I'll just colour in orange, okay? And now what's going to happen is these two stalks are going to fuse in the middle, okay? And the actual fusion event, what happens to the tip cells, is still an area of research. But what overall is going to happen is the two capillary buds, these two stalks, are going to fuse together to make one long vessel that joins these two capillaries, and hence you have created a new capillary through which blood can flow, and that should deal with the hypoxic problem that that area has. Okay, right, so that now completes our discussion of the ischemia-provoked angiogenesis pathway. Okay, what we will turn our attention to in the next video is the beginning of discussing angiotherapy. Okay, and we'll turn our attention firstly to angiotherapy for cancer. So we'll talk about carcinogenesis and how angiogenesis was seen as a potential target for treating cancer, and the slightly disappointing reality of the drug that we now have that can inhibit angiogenesis and how it hasn't had fantastic success in treating cancer. It does prolong life by a few months. It is horrendously expensive, but it does prolong life, but it's not doing the utter wonders we hoped it would do. Okay, and the reason for that we'll discuss in the next video in more detail, but it's probably because we're giving it maybe too late, basically. It might be that the tumours have already got their blood vessel network by the time we're actually giving the drug. Okay, and we'd need to be giving it long before you ever had any symptoms for cancer to actually stop the development of cancer. But then of course there's the side effects of taking the drug that mean that it's intolerable to give as a preventative therapy. Okay, but we'll discuss that in the next video.